A major advantage of the single living fiber that we study here, this intact muscle fiber system, is that we can study the contribution of calcium, myoplasmic free calcium concentration to force, calcium being a major regulator of force generation. So this technique is really important to that, and it being a living fiber, we can actually fatigue the fiber. That's a major point of our research, is to induce fatigue. So we can fatigue the fiber, it can recover. We can also put like pharmacological drugs or change the nutritional intervention, glucose, whatever we infuse onto the fiber, and look at how that affects fatigue or recovery processes in real time within the same fiber. And we can do these experiments over hours, if not days, technically within the same fiber. So I think there's a real strength to the technique, and also with that one fiber, we can also calculate the specific force and the force per cross-sectional area. The major advantage of the single fiber compared to looking at whole muscles is that in whole muscles you have hundreds, if not thousands, of fibers, and all of them are going to be activated and recruited at different time points, and it's hard to know what's happening in the muscle in humans in particular with what's happening with fatigue because of these activations and deactivations of fibers. Whereas when we have one fiber and we fatigue that fiber, we can really understand what is exactly happening with the force generation and how calcium contributes to that force generation over time, like in fatigue. And it's also a very powerful tool to also utilize in looking at disease as well, and muscle weakness. So with the intact single fiber method, it is manually dissected, and so this is used with forceps and fine tools, and pretty much what we do is the dissection procedure. You isolate the whole muscle, and you pretty much just cut away all the fibers that are not needed for your experiment until you have one. So a single muscle fiber is about the diameter of a human hair, so you can imagine this is a very tricky technique to learn, but it's a very powerful tool, and it's relatively straightforward once people gain the expertise. Equipment that we have here from Aurora Scientific with the intact fiber system, what is really nice about it is you have a force transducer that is accurate to measure the force ranges that we expect for an intact single fiber. And also on the other side, we have a motor that allows us to, it can be held in a static position to measure isometric force, but the motor can be moved to measure shortening and also lengthening, so we can measure power and velocity in the same fiber. Another really nice thing is there's also, of course, platinum plates that allow the fiber to be electrically stimulated, as well with a little chamber for superfusion of oxygenated tyrode to keep the fiber alive, or delivery of any nutrients or pharmacological drugs. And then there's also temperature control as well to try and keep the muscle at physiological temperature if that's the experiment that you want, or alternatively, we can, if you want to manipulate muscle temperature, we can raise the temperature or lower the temperature if that's a research question that you have. So that's a really great tool that we have that's connected then to a calcium measurement system that we have from Horiba. Equipment that we have, and with the goals of my research, is that we really want to understand the fundamental mechanisms that are causing fatigue under different types of exercise, and also understanding fatigue or weakness in disease as well. So this is kind of how this tool will be very useful in understanding the contribution of intracellular processes like calcium to the understanding of force generation.